Yeah. All right, so we're just gonna move on to chapter 10, Introduction to Simple Experiments. Okay, so a little chapter overview to start. So there's gonna be 2 examples of simple experiments we're gonna go through. Again, I'm literally just following what the book, says. So your book gives these 2 examples, and then throughout the chapter, it goes back and like refers to these examples. And like I've said for every single chapter, I would highly recommend that you read the book. The book is very good. It does a great job of explaining stuff. So definitely please be reading your textbook. If you haven't already for any of the chapters, now is the time to start. But anyway, we're going to have 2 examples, talk about different experimental variables, how experiments can support causal claims. So we're going to go back to those 3 causal criteria that we've talked about like 20 times already, but we're going to talk about them again. Independent group designs, within group designs, and then interrogating causal claims with the 4 validities, which again, all of the validity stuff should be a review by now. And hopefully by the end of this, you guys have all of that down pretty good. All right, so the 2 examples. The first example your book talks about is looking at the effect of different style of note-taking on test performance. So the researchers here divided students who are participating in the study, there's 67 of them, into 2 different groups. One group was instructed to take notes on a laptop, and then the other group was told to take notes just like with paper and pen, longhand. And the researchers thought that taking notes with like paper and pencil, longhand version, would lead to better test performance, especially on more conceptual type questions. Researchers thought that on laptops, listening to a lecture, whatever, students would just kind of try to type verbatim what was being said. It's easy to type fast. But if you're doing longhand note taking, students are going to have to kind of summarize, maybe draw pictures, write stuff in their own words, because it takes longer to write than it does to type typically. So they thought that that longhand method of note taking would lead students to actually process the information a little bit better, and then therefore lead to better performance on conceptual questions. So anyway, the students went into the lab, and then were told to watch a TED Talk picked beforehand on some sort of interesting topic. So the students were told to take notes on the assigned method. So sometimes there's computers in the rooms for the students to use. Other times there was pen and paper for the students to take notes that way. After the TED Talk, students had 30 minutes to do a different activity, trying to distract them from what they just saw. And then after the activity, they were tested on what they learned from the TED Talk. So the test that the students took had both factual questions, like asking what year something happened or something like that from the TED Talk, and then also conceptual questions that were more in depth, asking more conceptual type information. So these are the results of the study. The factual questions, Stella's in the background again. I'm sorry if she's loud. She's playing. For the factual questions, there wasn't a significant difference between groups on how they performed. But for conceptual questions, you can see longhand note-taking method, the old pen and paper. Students did significantly better on those types of questions than students who took notes on the lab. So in the coming slides, we'll talk about the different parts of the experiments, different variables, that type of thing, and we're going to refer to this example, as well as the second example, which looked at the effect of serving bowl size on amount of pasta eaten. So in this study, again, researchers divided participants into 2 groups. And then they went to the kitchen and were instructed to serve themselves pasta from a bowl. And the variable that they were looking at here, the researchers, they wanted to see if the size of the serving bowl affected how much pasta people took and how much pasta people ate. So they had a medium bowl condition and a large bowl condition. And then after individuals served themselves, the plate was weighed to see how much pasta they took. And then after participants ate, the plate was weighed again to see how much pasta each individual actually ate. So here are the results from that. So looking at just the serving bowl size, how much pasta like each group took. In the medium bowl condition, clearly individuals took a lot less pasta than in the large bowl condition. So how much pasta they physically put on their own plate. If they were taking it from a large bowl, they took significantly more than from a medium bowl. And then again, after the individuals ate the pasta was weighed again to see how much the participants actually ate, and that was translated into the amount of calories each group ate. And again, this is going to be a mean. I don't know if I said that, but the average for all the participants in each group. So in the medium bowl condition, they ate significantly fewer calories than in the large bowl condition. So not only did they serve themselves more pasta out of the large bowl, but that actually translated to them eating significantly more pasta, more calories in that condition, which is interesting. All right, so those are the two examples that we're gonna see throughout this chapter. So, all right, so what exactly is an experiment? An experiment, we've talked about experimental designs, experimental setups, like getting data empirically. So an experiment, you're gonna have at least one variable that's gonna be manipulated and one variable that is gonna be measured by the researcher. So back in one of the first chapters, we talked about different types of manipulated or measured variables. Talk about ethical ways of manipulating certain variables. In some instances, we can't manipulate variables. And then different ways to measure variables. Remember, we talked about self-report measures, behavior measures, physiological measures, right? So all that stuff that we talked about in previous chapters coming back into play here. In an experimental design, though, the manipulated and measured variables have fancy names. Not fancy, these you probably have heard a million times before. But the manipulated variable is also known as the independent variable. So the independent variable is the one that the researcher is going to assign participants to a different level or different, condition. So for example, that note-taking experiment, the independent variable is how the individuals are taking notes. So one group was told to take notes freehand, one group was told to take notes on the computer. So that variable, right, the method of taking notes is what's being manipulated in that experiment. In the pasta example, the size of the pasta serving bowl is the manipulated or the independent variable, right? Yeah. And another name for the different levels of the independent variables are the conditions. So in all cases, you're going to have at least two conditions. So in both examples, we had two conditions, either computer or longhand note-taking or medium bowl or large bowl of pasta. So those are the different conditions of the independent variable. So you at least need to have two, right? So you have, something to compare your groups, right? But you can have more than two. So if the pasta experiment wanted to have a small, medium, large, extra large bowl, whatever, right? You could have more than just two conditions, but at minimum you have to have two. So that's a manipulated or independent variable. Those are kind of interchangeable in this case. And then what we're measuring, the measured variable, is also known as the dependent variable, right? The dependent variable depends on our independent. We're changing this independent variable around and seeing how it's going to affect this, right? We're just measuring the change of the dependent variable. So I always think the dependent depends on the independent. That's how I keep them straight. But again, the dependent variable, the measured variable, you're measuring some sort of behavior, physiological measure. You can measure, self-report measures. But in all cases, you're measuring the dependent variable. So in the pasta experiment, you're measuring how much pasta they serve themselves, as well as measuring how much pasta they eat, right? So you can have more than one measured variable. And in the test-taking example, or the note-taking example, you're measuring how well they perform on those tests after note-taking. And they had the different, levels of different dependent variables they're measuring, just factual questions, and then the more conceptual questions. So you can measure a different number of dependent variables. but it's always going to be what you're measuring, right? Okay. Besides your manipulated or independent and measured and dependent variables, you also are going to have what are called controls or control variables in an experiment. And these are just going to be things that are held constant. So obviously, in an experimental setup, the whole point of kind of doing an experiment is to establish causation. And for causation to happen, we need to have really good internal validity, which we'll talk about later on. But control variables allow us to control for other confounds that could potentially be affecting our results. And a control variable is a misnomer because variables suggest change, except control variables are actually held constant. So it's not really a variable at all, but that's just what it's called. So for example, in the Posttest study, we wanted to make sure that the results, the dependent variable that we're measuring, the changes in that were actually due to our change in our independent variable, the size of the serving bowl, right? So we want to make sure that everything else is held constant so that the changes that we see are actually a reflection of what we want them to be. So the type of pasta in the bowl was constant between the conditions, right? We don't want the type of pasta to influence how much people serve themselves. The serving spoon, how big the spoon was held constant. Each individual's plate that they were, dishing their own pasta onto was constant. Everything else besides what we wanted to be affecting our dependent variable was held constant to ensure that what we saw, the changes we saw, were actually due to what we wanted them to be due to. Does that make sense? So control variables, everything really in experiments are going to be controlled for. If you don't control for them, you have, different things influencing your results. That's not a very good experiment. And you're never going to know if the results you saw were due to what you were manipulating or if it was due to something else, a different confound. And the whole point of experiments is to establish that causation and get really good internal validity and control for things that we want to control for. So those are control variables. Okay. So experiments supporting causal claims. We already talked about the criteria for causation multiple times, but you need to establish covariance, right? Just showing that the two things are related. Establish temporal precedence, so make sure that the variable you're manipulating is coming before, that change is coming before the variable you're measuring. So independent comes before dependent. And then also you want to establish internal validity, which I just kind of talked about, but making sure you're controlling for any confounds so there's not any alternate explanations that are causing the changes that you're seeing. Right, so independent variables answer the question compared to what, right? So you are always going to have a comparison group, at least two, I forget the word, two, I was on the first slide, two conditions, excuse me, two conditions at least, right? So you're comparing the different conditions to each other, right? You're changing an independent variable so you can compare it to something else, right? So you have a comparison group, comparison condition. And covariance is also not only just about the different conditions, but it's about the results. Not only do you need to have, two groups to compare to each other, but the results, you need to see a difference in what you're measuring to show that there's some sort of relationship between the groups that you're looking at, right? So not only about the independent variable and having your groups, but about the results, you need to see some sort of change between the groups to show that there's covariance, right? And you're going to have, in most cases, not all cases, a control group, which would have no treatment condition. Treatment groups, which is either just one or more treatment condition. And then in a lot of studies dealing with like drugs or some sort of pharmacological, experiment, oftentimes a control group is called a placebo group. So the placebo group, you've all heard of placebo drugs, is just like a sugar pill or something. The participants think that they're getting a drug or taking something. They don't know, exactly what it is, but they still have the idea that they're taking a pill, even though they're not actually getting any of the medication or whatever. So you don't always need a no treatment condition. So in like the plate size affecting how much pasta people eat, you're not really going to have a control group there, right? You're not going to have people who don't eat pasta because that's not going to help anything, right? So in some cases you don't necessarily need a control group, but in some cases you will. So that all should be kind of reviewed from previous classes. But the second thing, right, so we have covariance, just showing that our variables are going to be related. And then you want to establish temporal precedence. So we want to make sure that the cause variable is preceding the effect variable. So in the pasta study, experimenters manipulated the bowl size, which is the independent variable, and then they measured the dependent variable, how much people ate or how much people served themselves, to ensure that the causal variable precedes the effect variable in time. So just the nature of setting up experiments, we're manipulating one variable. Typically, we're manipulating that before we measure something. So it's establishing that temporal precedence, showing that, the independent variable is being changed, which is therefore affecting the dependent variable. So experiments, just the way that they're set up, do a really good job at establishing temporal precedence. And then the final bit of causality we want to look at is internal validity, right? So these are just the subheadings that we're going to talk about, but we'll talk about confounds, selection effects, design confounds, and then just confounds in general. We've talked about, how many times things that are affecting the study that you don't necessarily want to be affecting the study might be the reason you're seeing the results you are due to some other third variable, whatever it may be. So an example is if in the pasta bowls, the medium bowl maybe had a less appetizing pasta, the large bowl had a more appetizing pasta, right? If we're not controlling for the type of pasta in the bowl, then we're adding that variable that should be a control, but introducing a confound into the experiment. So maybe people ate more out of the large bowl because there was a yummier pasta in it. So we want to make sure to establish internal validity that we're minimizing all these confounds, controlling for things that we don't want to be affecting our results. So a design confound is when a second variable varies systematically along with the independent variable and can provide an alternate explanation for the results. So when you're setting up an experiment, if you have something else that changes along with your independent variable consistently, then it's a design confound, right? And we want to, it's only a confound if it's changing systematically, which means that when your independent variable changes, whatever this third variable is, it's going to change as well. So for that yummy pasta and not yummy pasta, if in the medium bowl condition, we always have a less appetizing pasta, then the large bowl condition has a yummier pasta, right? And if in all experimental setups, we have that type of pasta changing along with independent variable, then that might be an alternate explanation for your results, right? So if we have like a bunch of different experimental setups and the type of pasta is just completely random, sometimes the medium bowl is yummier, sometimes the other bowl is yummier. If it doesn't change systematically with the size of the bowl, that's not necessarily a design confound because it's unsystematic, right? So in not all experiments, but in some experiments, we're going to have some unsystematic variables, who the participants are that we can't necessarily control exactly for. But if it's unsystematic, so it's not changing every single time along with the independent variable, then it's not necessarily a problem. Only when we have a confound that's systematic in variability is there a problem and an actual confound in the experiment. So unsystematic variability, not a problem. It's just kind of random or haphazard, and it affects both groups. The book does a really good job of explaining this. If I'm not explaining it very well, there's like paragraphs and pictures and stuff in your book that kind of explain this. So read that. It's kind of hard to explain. I don't know. Read your book. Okay. But a design confound is when another variable varies systematically with your independent variable. Okay. Okay. We also have what's known as selection effects. A selection effect occurs in an experiment when the participants in one level of the independent variable are systematically different than the participants in the other level. So we can avoid selection effects with random assignment. We talked about this in previous chapters as well. You want to make sure that you're randomly assigning people to groups and not basing, who's in which group by some other variable. So again, if it's systematically different, that's a problem. If it's unsystematic, it's going to be random assignment. So an example, random assignment, just a definition of that, is a way to assign participants to levels of the independent variable such that each participant has an equal chance of being in each group. So you want to make sure there's no systematic difference between groups. And again, you're going to see the systematic difference or unsystematic multiple times. So make sure you get those two straight. But we want to make sure that everyone has an equal chance of being in each group. So this is kind of, we talked about random selection when we talked about external validity. Internal validity, random assignments, really important. Okay. So an example your book gives, there was a study done looking at the effect of an intensive treatment for autism or just regular treatment for autism. In the intensive treatments, the participants went to like a 40-hour-a-week treatment, and then they measured the improvement of each individual in just regular treatment or the intensive treatment condition. So for this study to actually work, to see if there's good internal validity, you want to make all the participants have equal chances of being in just regular treatment or the intensive treatment group. But the study, because you're having different treatment conditions, it's not really ethical to, not allow certain people in the intensive treatment group. They allowed the participants to self-select. And what they saw is that the intensive treatment group had better improvement than just regular treatment. But because the participants self-selected, it's not clear that the actual treatment is what's causing the improvement, right? Because if people who selected to be in this intensive 40-hour treatment group, maybe the parents were driving the kids to the treatment. People who, worked more couldn't do that, so they had to be in the regular treatment group. People in this group are just, maybe more willing to, do more for their kids, try to help their kids. So they wanted them in this group, right? So we're not necessarily sure that the treatment itself is what's causing the improvement. To do that, you would have to have complete random assignment to make sure that participants in both groups are equally represented. Okay. Another way we can avoid selection effects is to do something called matched groups or matching. So if we have 30 people, we want to see, how meditation improves their test scores or something. When dividing them into groups, we're first going to match them up and then randomly assign them groups. So you're going to match groups with some variable that's important to your experiment. So if you're trying to look at like test scores, you might match people up based on GPA. So first you're going to find the 2 highest GPA individuals, match them up, and then randomly assign each person to a group. So not necessarily 1 and 2, but just, randomly assign the top 2 people to a group. The next 2 highest GPAs, again, match them up and then randomly assign them to a group, and so on and so forth until you get to the lowest GPAs, match them up, and then assign them to a group. Obviously, you need an even number of participants for this type of study or this type of grouping, but this is good because it's still random assignment, because you don't necessarily, you're not picking which person is in each group. So within the groups, it's random who's going to be there, but you're just making sure that a variable that's important, such as your IQ or GPA, whatever it might be, making sure that the participants are kind of evenly distributed between groups. If you were just take all 30 people, put them in a pool, and then randomly select who goes to which group, you might have one group that has, on average, a higher GPA than the second group, just even if it's random. So this way you're kind of controlling, making sure that the GPA averages in each group are kind of consistent. So that's called match grouping. And it just helps, again, avoid selection effects. In some cases, you can't necessarily do this, but in other cases you can. So good thing to consider. Okay. So now we're going to talk about different types of group designs. So independent group designs and within group designs. So the first here is independent group designs. Look at post-test only, pre-test, post-test design, and then which is better. So independent groups versus within groups. What's the difference? So independent group designs are also known as between subject design or between group design. And in this type of design, in each level of the independent variable, each condition that you're creating, you're going to have a different selection of participants. So in the pasta example, one group that was randomly selected did the medium condition, one group that was randomly selected did the large condition, right? But each condition had a different set of individuals participating. Okay, within group designs or within subject design, each participant is presented with all levels of the independent variable. So if you conducted a note-taking study and each participant did both longhand and laptop note-taking, that would be within group design. In the study that we actually talked about, they divided the groups. One group did longhand, one group did laptop note-taking. So that was technically independent, right? Because they were in separate groups, separate participants for each control, I always forget that freaking word. It's a C word. I said it just 2 minutes ago. Each level of the independent variable, right? You have two separate groups doing two separate things. Within groups design, all the participants would be exposed to both of the levels of the independent variable. Okay, and we'll talk about more in depth these in a second. So for an independent desktop group design between subject, we have two different groups. There's different ways you can do it. So the first is a post-test only. And again, it's an independent group experiment. So the participants are divided into two different groups, and they're only tested on the dependent variable one time, right? They have one post-test. So you randomly assign people to group 1 or group 2, and then measure the dependent variable after they do whatever they're supposed to be doing. Okay, so for our example, randomly assigned people, one group did laptop notes, one group did longhand notes, and then they took the comprehension test, and their comprehension of the material was measured at the very end of the experiment, right? That's a post-test design. So they're only given a test for the dependent variable one time, and it was after they took the notes, right? Post-test. And again, independent group, because each group only was exposed to one condition. Okay. There's also something known as the pre-test, post-test design. So again, you randomly assign people to two different groups. So in this case, you're looking at mindfulness and nutrition, taking each of these classes, what effect that has on the GRE score. But in a pre-test, post-test, individuals are given a test before they're exposed to the independent variable, and then again after they're exposed, right? So in this case, people were assigned to either take a mindfulness class or a nutrition class, and researchers wanted to see what effect taking the class would have on the GRE score. But before they even took the class, they took the GRE the verbal GRE section, and each group had an average score. Then they took the class, and then they were tested again to see how the class affected their score from before. So that would be a pre-test and post-test, right? So figuring out the baseline score for each group before they were even exposed to whatever the class was. Does that make sense? So this was actually a study that was done. This are the results from it. So the the white bars here was the pre-test scores on the verbal GRE. So you could see at the start, the nutrition group and the mindfulness group, before they were exposed to their experimental condition, they scored pretty similarly, right? The average of the scores was pretty much equal. And then they were exposed to the class, and then they took a post-test, and you could see the accuracy and the nutrition class decreased, mindfulness class increased. Okay, so on the second measure, you're comparing it to that initial measure. And the initial measure is just kind of to make sure that each group has kind of equal starting ground. They don't have, this randomly assigned group was just better to begin with, right? You want to make sure that at the start, you're kind of, each group has, equal chances of doing well on the exam. Okay, so which design is better? This slide just says nothing, basically. Basically, in some situations, it's better to use just a post-test. Some situations, it's better to do a pre-test, post-test. It really depends on your experimental design. Post-test only, even though it might intuitively might think that a pre-test, post-test would be better to see, changes. Post-test only can be just as powerful as a pre-test, post-test. In some cases, you can't necessarily do pre-test, post-test. If you are having your participants eat, you can't do a pre-test, post-test, because after they do a pre-test and you see how much they eat normally, then they're going to be full, and then you can't see how much you're going to eat after whatever the experiment is looking at, right? Or if you're doing some sort of like workout experiment, if you do a pre-test, again, they're going to be tired. So you not necessarily a good idea to do a pre-test. Okay, so what design's better? It depends on the situation. Independent group design. Within group design, there's two different types: concurrent measures and repeated measures design. We're going to talk about some advantages, talk about establishing, causal criteria, disadvantages, and looking at pre-test, post-test within groups. Okay. So the first type of within group design is the repeated measure design. And participants are measured on the dependent variable more than once. Okay. The example of this, researchers theorized that when experiencing something, whether it's eating chocolate or viewing a painting or something sad, eating something that you don't like, if you experience that alone, your emotions are not going to be as intense as you experience it with someone else, right? So looking at shared experience, emotions, perceptions of things versus unshared experience. So what they did, they had participants come in, and they, so there's 23 college women. Each participant was joined by a female confederate. They sat side by side. They never spoke to each other, talked to each other, looked at each other, anything. And cards were flipped up, and it said like what they were supposed to do. So some of them were eat chocolate, some of them were look at a painting, whatever. They were told the cards were random. Each of the two people were doing something different, right? They had different cards, but the cards actually weren't random. So they had, when the first card was flipped up, it's told both individuals to taste a chocolate, and both individuals got that same card, so they tasted the chocolate together. After they tasted the chocolate, they rated the experience of the chocolate. Did they like the chocolate? How good was the chocolate? And then another card was flipped up. The individual that was actually in the study was told to taste a different chocolate. And then the other participants, the confederate, was told to look at a painting or something. So then they tasted the chocolate alone and was told to rate that chocolate again. Right? So seeing if you're tasting something with someone else sitting next to you, does that affect your rating compared to tasting something on your own? So the individual was told that these were different chocolates, right? But they're actually the same exact type of chocolate. So the rating theoretically should have been the same, right? It's the same thing that they're tasting. Right? Repeated measures, they are exposed to, they're measured on the dependent variable more than once, right? So they're exposed to one thing, rate it, exposed to the other one, and then rate it again, right? Two different dependent variables exposed to at different times. And researchers found that when they ate the chocolate with someone sitting next to them, they rated it, who was also eating the chocolate, they rated it as more tasty than when they ate the chocolate alone. Okay, so repeated measure, again, you're exposed to one condition, exposed to the second condition, third condition, however many conditions they might be. But yeah, and again, because it's a within group design, each participant is exposed to all of the different independent variables. Yeah? Okay. Again, your book explains this really well, so read your book. The other type of design is concurrent measure design, which participants are exposed to all of the independent variable levels, conditions at the same time. And the dependent variable is measuring like the preference. So the example on this slide is looking at preference in infant research. So what they did, basically, they had babies sit or whatever, look at a screen, and on the screen at the same time, there's a female face being shown and a male face being shown. And then they just looked at the preference of which face the baby preferred to look at, right? So concurrent measures, both of the independent variables were exposed at the same time, and the dependent variable is just which one is preferred. So yeah, you're not like showing the baby the female face first and telling them to rate it, and then the male face and then rate it again. That would be the repeated measure design if you're showing one thing after another. But if you show both at the same time and look for preference, that's concurrent measure design. Okay, does that make sense? Any questions so far? No? Okay, good job. All right. Some advantages of within group designs. It does a good job of making sure that participants in your group, there's no issues with your groups being different systematically, because everyone's participating, doing everything, right? So you know for sure that maybe if you're doing that chocolate study, there's not an external factor of maybe one group just likes chocolate better than the other group, right? If everyone's doing the same thing, personal preference isn't playing a role. It's easier to notice difference between conditions, and also you need fewer participants generally, right? Because if you're doing an independent design, you have two conditions, different people in each condition. Say you have 20 people in each condition, you need two different groups of 20 people, so 40 people total. If you're doing within groups design, the same people that did your first condition are also doing your second condition. So if you're doing, again, 20 people in each condition, it's the same 20 people, so you just need 20 people. So it can save on cost, that type of thing, if you do within group designs. Okay. Looking at covariance, temporal precedence, and internal validity in within group designs. So these are like the section headings. So order effects, some practice effects, and carryover effects. And how to avoid these different order effects with something called counterbalancing. That's what we're going to be looking at. Okay, so order effects. When being exposed to a condition, it can affect how participants respond to other conditions. The first type of order effect is called a practice effect. So participants either get better at a task from practice or get worse at a task due to fatigue. So fatigue is actually called fatigue effect. Right, so if you have someone do some sort of task multiple times and you're trying to see how the color of the room or something is affecting how well participants do on a maze, you give them the same maze 5 times and just place them in different rooms, by the end of the experiment, they're going to be so good at that maze regardless of what color the room that they're in, right? They've been practicing that. They're better at it. So at that point, you don't know necessarily that the color of the room is actually what's influencing how well they are doing on the maze. It's just, they're good at it because they practice it. Or fatigue, if you're doing some sort of workout, something like that. Again, if they're doing something repeated amount of times, after a while they might get tired of doing it, so they get worse, not necessarily due to any change in the independent variable. Another type of effect is called carryover effect, which is a contamination carrying over from one condition to the next. So if you are having people taste something, you have them taste a mint first and then taste the orange juice or something next, right? That doesn't taste good. Their mouth still has a mint taste to it, so they're not gonna like the orange juice regardless of if they would like it, otherwise, because they have a carryover effect from your mint, right? Let's see what my notes say for another example. You drink caffeinated coffee and then take a test. You then drink decaf coffee and take a different test. You would have carryover effect because that caffeinated coffee is still in your system, so it's affecting your results on the second test, even though you drank decaf afterwards, right? So just contamination from the first thing to the next. Pretty straightforward. Ways to avoid this is something called counterbalancing. So presenting the different conditions in different orders. So you can do full counterbalancing, which you have all possible condition orders presented. Or you can have partial counterbalancing when only some of the possible conditions are used. So if you don't have many different conditions, it's easier to do full counterbalancing. If you have two things, taste chocolate alone, I'll just go to the next slide. Taste chocolate alone, taste chocolate, with someone sitting there. It's pretty easy to do a full counterbalancing condition because one group tastes it alone and then with someone else, and then the other group tastes it with someone else and then alone, right? You're doing all possible conditions here, counterbalancing, to make sure that it's not just, the first time you eat chocolate, it tastes better, and then the second time. But if you have more than two conditions, there's the word. If you have more than two conditions, the amount of, different assignments, which one comes first, second, and third, fourth, fifth, sixth, right? As we increase the number of conditions, it increases the number of ways that the experiments can be set up, which is sometimes hard to do. You don't necessarily need to have a full counterbalance. You can have that partial counterbalance. So only some of the possible conditions orders are used. There's something called the Latin square, which just gives a randomized order for a participant where each condition appears at each position at least once. That, look in your book, it gives you a good example. But you want to make sure that you're controlling for these things that can affect, your results, and counterbalancing is a way to do that. Okay. Within group designs, some disadvantages. Again, there can be order effects. So if you have, everyone being exposed to all different levels of the independent variable, it's easy for those, the order effects to influence the results. So if someone, okay, I don't need to give more examples. Potential for order effects, but we do have a solution, which is counterbalancing. It just can be difficult in some situations to adequately counterbalance. Might not be practical or possible. So if you're trying to see which is the best way to teach someone to ride a bike, you can't have a child learn to ride a bike one way and then the next week teach them the second way and try to figure out which way is better, because after you teach them once, they already have learned something, right? So in some situations where you're testing learning, after one exposure to one condition, it's not you're not able to accurately measure exposure to the second because they've already been exposed to one, which is going to influence, no matter how you set up the experiment, the second condition. So in some cases, it's not practical. And also sometimes if you're experiencing all levels of the independent variable, it can change the way participants act. This is known as demand characteristics. So for example, if you have the participants, seeing how much pasta they take from a medium bowl or a large bowl, and you have them take it from a medium bowl first and then a large bowl second, it might be pretty easy for them to figure out, what's going on. They know they're in an experiment, know something's happening. So if they see that there's different sized bowls, that's the only thing that's changing, then they know that this has something to do with that, and they can change the way they act because they know what's going on, right? So demand characteristics, not always good. Is a pretest post-test a repeated measure design? Answer is no. Pretest post-test, you're measuring the dependent variable more than once, yes. But in a repeated measure design, you have exposure to all levels of the independent variable, right? So in pretest post-test, even though we're measuring dependent variable more than once, one group is only exposed to one independent variable, the other group is only exposed to the second independent variable. To be a repeated measure design, you have to have both groups being exposed to both variables. Okay, so make sure you have these straight. Know which one is which. You might see a little matching a quiz or exam. Okay. And then the last bit here, review. This is a long lecture, I'm sorry. Interrogating causal claims with the 4 validities. We've talked about these so many times. Should be well ingrained in your head by now, but construct validity, looking at how well the variables were measured as well as manipulated. External validity, who can the results generalize to? Statistical validity, how well does the data support the conclusion? Is the causal claim actually supported by the data we collected? And internal validity, making sure there's no confounds or alternative explanations for the outcome. So just a little bit more on each. Construct validity. So since we have a dependent and independent variable, we need to not only look at how well dependent variables were measured, but how well independent variables were manipulated, right? So there's a thing called a manipulation check, which is a way for researchers to gauge how well their independent variable has been manipulated. So let's see, my example, I forget. I forget the example. Oh, shoot. Let's think. Manipulation check. How well, let me see. Hold on. Okay, example. Researchers wanted to investigate if humor in lectures would improve retention of the material. So they had individuals listen to boring, not humorous lectures versus like what they thought were to be more humorous lectures, and then saw the effect on their exam scores. But they did a manipulation check. So besides just measuring the exam scores, they also asked students how funny or humorous they thought the lecture was. So that way they could actually, the researchers actually could rate how funny or humorous, the students thought each lecture was, to make sure that they're actually manipulating that humor level of each lecture. Okay, so you could do a manipulation check, which is like within the study that you're doing. You could also do a pilot study, which is just a simple study that is completed before or sometimes after, but making sure that you're manipulating the variables that you want to be manipulating. So you could do it the same example. Again, you want to have students listen to different lectures. Some are funny, some are not as funny, see how it affects their exam scores. So you could have, before the actual experiment, have people listen to different lectures and just simply rate them on how funny they were, or how humorous, whatever. So your pilot study is just to determine, how well you're manipulating the variables, if the lectures are actually different in terms of humor. So then once you have that established and know for sure that you manipulated the variables correctly, then you'll do the actual study and see, in each condition, did the students learn better or worse? Okay, so that's just making sure that you're manipulating the variables correctly. Okay, external validity, seeing to whom or what the causal claim can generalize to. So we've talked about, in most cases for the external validity, generalizing to other people. So looking at how participants were selected, did you use some random sampling technique? Was your population or sample of interest unbiased? Making sure that you can generalize what your research found to other people. In most cases, in a lot of the studies we talked about, at least, the participants were all college students who wanted to participate in the study, so it wasn't necessarily random sampling. It could have been just convenient sampling. So not necessarily the best external validity. Because in most cases, in experimental setups, internal validity is a bit more important than external validity. It's more important to have a good experimental design and setup, not necessarily worry too much about who's participating, right? If they're randomly assigned to groups, ideally you would think that, you could generalize it, but in most cases, internal validity for experiments is more important than external validity. But besides generalizing to other people, to the population, you can also generalize to other situations. So trying to figure out when we're doing experimental setups. So for example, in the note taking, they watch TED Talks, right? So what if you're in an actual lecture watching a live professor? Is that going to be different than watching the TED Talk? What if we have younger students versus older students? That's kind of people, but right, there's different situations that we can think about too. What type of class it is, right? Is it a anatomy and physiology class where it's more factual information versus some philosophy class where it's more applying, right? So depending on the situation, the results might not always be generalizable. So just important to think about. And if external validity is poor, we talked about this previously as well, that doesn't mean the whole study is trash, right? In most cases in science, you're going to have multiple studies looking at just small samples of people that might not generalize to everyone, but there's enough studies that kind of do the same thing with different groups of people that it can eventually kind of generalize to more situations or more people. You're not going to have one study that's going to be able to generalize to every single person in every single situation. It's just not plausible. So in most cases, experimental setups, internal validity is more important, and you can take multiple studies that have different samples of people in them, different situations, so that we can generalize to more. Okay, and then statistical validity. How well does the data support the causal claim? So again, we've talked about this previously, looking at if the differences between groups is statistically significant and how large the effect size is. I'm not going to get too much into that. Read your book. You took a statistics class already. And then finally, internal validity, we talked about a lot, but making sure that there's no confounds. And in an experimental setup, internal validity is the most important, right? So there's 3 fundamental questions about internal validity you want to ask. Is there any design confounds, right? So making sure that as the independent variable changed, we're not changing something else with it. If you have an independent group design, did the researcher control for selection effects using random assignment or random, matching? If you had a within group design, did the researchers control for order effects with counterbalancing? So these 3 things are really important when you're setting up an experimental design. Internal validity, again, is always going to be the most important thing when you have a causal claim and you're doing an experimental setup. Okay, So that was an hour long. I'm sorry. You can watch it. Well, it's the end now, but you can watch it in little sections or whatever. You're not going to hear this till the end, so that doesn't matter. Claire, great. Definitely read your textbook. If you have questions, let me know. Again, I'll post like a little question form for you guys to ask questions on if I haven't done it already. I don't know. I don't know what I've done yet. So, yeah. Okay. Good job, guys. You're so smart. You're doing great. Okay. Bye.